Hi there, my name is Dr. Douglas McBean, and in this lecture today we're going to be looking at the use of antibiotics, and we're going to be starting by doing a quick overview of terminology, get you used to sort of more familiar with that terminology, and then looking at the ways that you can actually get antibiotic or antibacterial action, and then we'll look at some specific examples. So starting off with the terminology, the main thing about the treatment of infection is the key here is about cell death, but what we're looking for hopefully is selective cell death. We're not just wanting to kill all the cells in the body, it's targeted very specifically at the bacterial cells, the microorganisms. So that's where the term antimicrobial comes in. An antimicrobial is an agent that will kill a microorganism, or if not kill it, will at least inhibit its growth. The term that we're probably more familiar with is antibiotic. Now, that was initially kept for substances that were produced by microorganisms that killed other microorganisms, or equally so, inhibited the growth. So what we had was an antimicrobial that was any agent that would kill a microorganism, and an antibiotic, which was a microorganism or a substance produced by a microorganism that would kill another microorganism. So we had the two terms, antimicrobial and antibiotic, which have now become pretty much interchangeable. And probably the one that's most familiar to most people is the use of the term antibiotic. Some other terminology that you need to get your head around is the use of the phrases bacteriostatic and bactericidal. So whether it doesn't really matter if we go back and see there, it doesn't matter whether we're referring to a drug as being antimicrobial or antibiotic. Within that, we have these classes. Because what we said was these drugs are either going to kill the cell, the microorganism, or they're going to inhibit the growth. If they're killing, they're bactericidal. If they're inhibiting the growth, they're bacteriostatic. Another phrase that you may have heard of is the spectrum of activity. And that is the number of different types of organism that are sensitive to a drug. So you might have heard of the use of what they refer to as a broad spectrum antibiotic. That means it targets a wide range of different bacteria. And then the flip side of that is a narrow spectrum antibiotic, where that's only going to be effective for very few species of bacteria. So obviously, if you're not sure what drug's going to work, a broad spectrum is more likely to have an impact on a wider range of bacteria. Another thing that's getting a lot of coverage at the moment is this idea of resistance to antibiotics. And this seems to be becoming a more and more prevalent problem, a bigger and bigger issue. And so therefore, what that has resulted in is there are a number of antibiotics that are no longer effective. They're no longer to either kill or suppress the growth of microorganisms. And so what that then can result in is microorganisms that are resistant or no longer susceptible to the drug. Now that resistance can come about in a variety of different ways, but it's the microorganism that's adapting. So it comes under attack from the antibiotic, and then if you like, it starts to think, how am I going to defend itself? Obviously not thinking, you know what I mean? But the microorganism is going to go, right, I'm under attack here, how can I alter my defences to minimise the impact of this attack? Now, that resistance can come about because the drug is obviously going to have to work on a receptor, and then the microorganism can alter the receptor target. The microorganism can limit or control the amount of drug that actually can cross the membrane and get in to do the damage. It can alter the metabolic pathways, and it can develop ways, therefore, to make the drug inactive. Another question that you might have heard, they use the term friendly bacteria, and some people get a bit confused as to what that means. So it does beg the question, are all bacteria disease causing? And the answer to that is no, they're not. More than 90% of the cells that are in the human body that are of prokaryotic origin would be referred to as normal flora. And infections from those normal flora would only occur when our defences are down. That is, when we're maybe immunosuppressed, for example. Or when the normal microorganisms, if you want to call them that, the good bacteria, translocate or move to a different location within the body. So what you want to do is, if you've not done this already, get yourself onto the BNF, the British National Formulary, and then when you do a search under the terms antibacterial or antibiotic, you will see the full list of drugs that are currently being prescribed within the UK. And there are 4 main mechanisms of action that these drugs have. Now, we're not going to go through them all. We're going to focus on 2 specific types that relate to the 2 most common antibiotic medications that you're likely to become involved with. So the ways you can do it, you can inhibit the cell wall synthesis, you can inhibit the protein synthesis of the microorganism, you can inhibit folic acid biosynthetic pathways, and you can inhibit the DNA RNA synthesis. And all of those will affect the way that the microorganism behaves and give you either a bactericidal effect or a bacteriostatic effect. The 2 that I've highlighted here in red, those are the 2 that we're going to focus in on, because they're the 2 classes that you're more likely to come into contact with. So we're going to focus in on inhibition of cell wall synthesis and inhibition of protein synthesis. So what we've got here is a little diagram showing you how all the different drugs work. Okay? And you'll see there that there's the cell wall synthesis of the microorganism, and you can have drugs that will work there. We've also got drugs that link in, the ones that are involved with DNA and RNA. We've got ones that are involved in protein synthesis and ones that are involved in folic acid synthesis within the cell. And if you so you see where the crosses are, these are the drugs that inhibit cell wall synthesis. These are the drugs that interfere with DNA and RNA. These are the ones that interfere with the protein synthesis, and these are the ones that inhibit folic acid. And as I said, we're going to focus in on cell wall and protein. So we're going to focus in on these drugs here that affect the synthesis of the cell wall, and these drugs over here that affect protein synthesis. So the cell wall synthesis inhibitors fall into a multitude of different drugs, but again, because there are so many, we've already said we're not going to look at all 4 classes, we're going to look at 2, and then within each class we're going to focus in on a particular subgroup within the class. And in this case, the cell wall synthesis inhibitors, we're going to focus in on the penicillins. Drugs like amoxicillin, flucloxacillin, etc. So these are the, if you like, the go-to guys. Amoxicillin in particular is one of those drugs that we mentioned before as being a broad-spectrum antibiotic. So, because these drugs inhibit the cell wall synthesis, the penicillins are bactericidal. They're going to kill the bacterial cell. They're not just going to inhibit its growth, they're going to kill it off. And that's why they're so effective, and that's why they're broad spectrum. What they do is they interfere with the formation of the peptidoglycan layer. So if the penicillin comes in, it binds to a transpeptide enzyme. And that transpeptide enzyme has a function which cross-links the muramic acid and the glucosamine. Now, that cross-linkage of muramic acid and glucosamine gives the bacterial structure stability. So, if the penicillin comes in and binds to that, it will impact on the muramic acid glucosamine cross-link, and therefore will interfere with the structural stability of the cell. Now, the transpeptidase that we mentioned, if I just think back, that transpeptidase enzyme here, the transpeptidases, that's one of several proteins that penicillin binds to. And because it's a protein, although the transpeptidase, the ASE ending at the end means it's an enzyme, but enzymes are of course proteins, excuse me, because this is one of several bacterial proteins, they're referred to collectively as the PBPs, the penicillin binding proteins. Now, other proteins, PBPs, that penicillin activates include the autolysins, and when you activate the autolysins, they cause the hydrolysis and destruction of the components within the cell. And the PBP, the penicillin, also activates these proteins, the carboxypeptidases and the endopeptidases, which break down the peptide bonds and therefore impact on the formation of proteins. And proteins can be structural and functional. So what you're really looking at then is the penicillins, if we just make back one, they're going to take away the structural stability of the bacterial cell. They're going to destroy the intracellular components, and they're going to break down the proteins and the structural and functional integrity of the job that they do. So therefore, that's why they're breaking down so many things that they're going to be bactericidal. Now what happens is, the penicillin comes in through a pore and crosses the outer cell membrane. And then it comes down, and there's the peptidoglycan that we mentioned. So what to do is keep referring back to, the various structures that we talked about, because we said they interfere with the formation of the peptidoglycan layer. So that layer there, that's blocked by penicillin, and so therefore it travels right through, and it goes to these binding proteins here that we talked about, the penicillin binding proteins, and that's where you get the effect once it actually binds onto the penicillin binding proteins. And that's what we're seeing here. There's the penicillin coming through the outer cell membrane, through the porin, the little gap for it to go through, passes here because this is the synthesis of this peptidoglycan layer is blocked, so it's not there to obstruct it. It moves through, comes in, and binds here. But you'll see there that there are also enzymes, the beta-lactamase enzymes, that will inactivate the penicillin. So as well as penicillin being able to bind here to produce the effects we've mentioned, it can also be deactivated by the presence of beta-lactamase. Now, we said that it's possible that the bacterial cell can become resistant to the penicillin. And now, obviously, to get into the cell, they have to cross that cell membrane. Now, the bacteria can become resistant to the penicillin, and it can do this by, the bacterial cells are like any other cells, they have the ability to make proteins, and they can alter the proteins that they make. So what they can do is they can actually modify the PBPs, the penicillin binding proteins, so that penicillin doesn't bind as well. The other thing that they start to do is they can develop the mechanisms in their cell membrane to pump the penicillin back out. They can also produce the beta-lactamases, or penicillinases as they're sometimes referred to. And what they do is they're an enzyme that breaks down the penicillin and inactivates the penicillin. And they can also alter the porins. So what we're saying then is, if you go back to our little diagram, the bacterial cell itself can change the structure here so the penicillin doesn't come in, and can increase the levels of this to break the penicillin down before it can bind, and also alter these so the penicillin doesn't bind as well. So those three mechanisms, 1 doesn't enter as well, 2 gets broken down more, 3 structural change with these proteins so they don't bind as well. All of those will render the penicillin either less effective or not effective. Now, penicillin, assuming it's still going to work, is the main broad-spectrum antibiotic of choice. But you can have a hypersensitivity to it, and that's the major adverse effect. So some people, when they get asked the question, is there anything you're allergic to? A number of people will be allergic to penicillin. I am myself. I can't take penicillin. So if in the past, if I've needed an antibiotic, I'm going to go with I'm usually given the next class that we're going to talk about because of the hypersensitivity. Now that can range from a mild thing, like you can get an itch, you can get hives, like a skin rash, you can get some respiratory distress like an allergic response, right through to full anaphylactic shock. Now, the other thing is the main route of excretion of penicillin is through the urine, and it's through the urine and it's unchanged, so it pretty much passes through in its pure form. You've got to be therefore very careful if you're giving a penicillin orally to a patient who has got renal insufficiency, because if the kidneys aren't working properly and they're not clearing the drug out effectively, then there's potential for the drug to build up to toxic levels. So if you're giving it to a renal insufficient patient, you will have to lower the dose accordingly. Sometimes it can be important to give an antibiotic intravenously to get the dose in there to fight the infection rapidly, get it straight into the circulation. You've got to be very careful there, because if you are giving a high IV dose, the penicillin can either cause seizures in some patients or have an antiplatelet effect, meaning it can alter the blood clotting capabilities of the drug itself. So you've got to be very careful with that as well, to make sure that you don't get the patient, cause them any main problems. Other things that can happen, it can cause anemia, it can cause thrombocytopenia, and it can cause hyperprothrombemia. So you can get difficulty with clotting of the blood. That's where the major issue comes from. The second class of agents are the ones that inhibit the protein synthesis. And again, we've picked out one subclass within it, which is probably the most common drug that's prescribed when a person can't have penicillin or penicillin doesn't work. And that's from the group of drugs known as macrolides or erythromycin. Now, the reason why that's the first line of defence in somebody that can't take penicillin is because the macrolides, erythromycin is the example, they have a similar broad spectrum of activity to the penicillins. Now, what they do is they inhibit protein synthesis by binding onto the ribosomal 50S subunit. And that then prevents translocation of the amino acids. So the amino acids can't come on and join up, if you like, into a chain to make a protein. So therefore the protein synthesis is switched down. Now, dependent on the concentration of these drugs, they can either be bacteriostatic or bactericidal. The higher the concentration, the more likely you are to see a bactericidal effect. So what they do is they bind onto the 50S ribosomal unit here. So that's the top part of this diagram on the messenger RNA there. If you look at it, you'll see the messenger RNA running through the middle. Then you've got the 50S and the 30S subunits. And so what that does is the incoming amino acids would normally come in and you would get the development of a chain, a long chain of amino acids, which would ultimately make the protein. Yeah. What they do is the drugs, if you like, interfere with the peptidoglycan transfer enzyme, which blocks the linkage of the incoming amino acids. So the amino acids don't come in, don't join up, and you don't get the same level of protein synthesis as you did before. Now, in the same way as we can get resistance to the penicillins, we can get resistance to erythromycin, the macrolides. Again, how? Well, same as we said before, the membrane of the bacteria will change to make it more difficult for the drug to enter the cell, the bacterial cell. That's the same here. The permeability of the bacterial cell membrane can be altered so that the macrolides don't enter to the same extent. They can then methylate the bacterial 50S subunits so that you don't get that same, so that the drug can bind to actually start to affect the messenger RNA. And then finally, the bacterial cell will develop new proteins which will work enzymatically to break down the macrolide drugs that are coming in. So why would you choose it? Well, as I say, if somebody's got a penicillin hypersensitivity, it's the go-to guy. It's the first drug of choice after that. But it's also importantly, it's actually is the number one choice, regardless of penicillin resistance or not, for certain infections like Legionella, Legionnaire's disease, or pneumonia. It's the systemic drug of choice for impetigo and also for community-acquired pneumonia. So, although it's sometimes considered that the macrolides like erythromycin are second choice antibiotics if you can't take penicillin, there are certain bacterial conditions, certain infections where they are the number one choice. Probably most important from a podiatric point of view, for example, the macrolides are used to treat soft tissue infection. They're also very effective for genital urinary infections, or urinary tract infections. They're good for treatment of acne, and they're actually very good in eye drops if someone's got an eye infection. But this idea of them being a great choice for a soft tissue infection is probably something that's probably most useful or of most interest to a person in a podiatric position. I've also put a little bit in here about Lyme disease, which with a link through to the Mayo Clinic. And the reason for that is it's something that's that comes up. It's something you read about. It's the one of these ones where it's heavily wooded areas and people get ticks carrying the disease. And erythromycin is particularly effective at dealing with that. We mentioned with penicillin that they do have adverse effects, the penicillins, and so do the macrolides. Probably the main one is gastric distress. So they can cause quite bad levels of gastric upset, flatulence, indigestion. A lot of people sometimes say that they don't really feel too bad, but they feel a little bit nauseous or a little bit like they can feel their stomach churning. So it's a little bit of discomfort in a lot of cases. But that can be minimised by taking it with food, and also the development of what they call gastro-resistant tablets. These are ones that if you take them with food, they actually in a lot of cases, they'll have a very limited impact on the gastric system. So they're trying to minimise that. The other thing you've got to watch out for is they inhibit certain P450 enzymes. And so therefore, you've got to be careful. There are certain drugs that are contraindicated. That is, you can't take a macrolide like erythromycin if you're on certain classes of drugs. And that's because they affect the enzyme that metabolises the other drug. And if they affect the enzyme that metabolises the other drug, then the other drug doesn't get metabolised properly and could build up to toxic levels. So you've got to be very well aware of that. But the British National Formulary, for example, does give you that level of information. Just to get a general overview of the topic area, to kind of summarise all of this, I've given you this link to the NHS pages, which take you through to a whole thing here about antibiotics. Yep. And it's just an interesting little overview, talking through when are they used, how do I take them, sort of patient information, considerations, interactions, how they interact with other medicines, that's down here, the different types. So we mentioned the penicillins, we've mentioned the macrolides, yeah, and you'll see there, useful for treatment for people with penicillin allergy. There's also a good section here on antibiotic resistance. So there's a whole lot of information there. If you fancy finding out a little bit more about it, that's the place you need to go. So in terms of a summary or an overview, what you need to get your head around is the basic terminology that we use, the idea of antibiotic resistance, and different ways that you can upset the bacterial growth. But we've focused in particularly on two main areas. We've looked at the penicillins and the macrolides. The main drugs being amoxicillin is a penicillin and erythromycin is a macrolide. And they're probably the two most commonly prescribed drugs in this area. Thank you very much.